Climax and post-climax. In certain areas, different climax communities develop under similar climatic conditions. If the community has life forms lower than those in the expected climatic climax, it is called preclimax. A community that has life forms higher than those in the expected climatic climax is postclimax. Preclimax strips develop in less moist and hotter areas, whereas postclimax strands develop in more moist and cooler areas than that of surrounding climate. Theories. There are three schools of interpretations explaining the climax concept. Monoclimax or climatic climax theory was advanced by Clements 1916, and recognizes only one climax whose characteristics are determined solely by climate, climatic climax. The processes of succession and modification of environment overcome the effects of differences in topography, parent material of the soil, and other factors. The whole area would be covered with uniform plant community. Communities other than the climax are related to it, and are recognized as subclimax, postclimax and disclimax. Polyclimax theory was advanced by Tansley 1935. It proposes that the climax vegetation of a region consists of more than one vegetation climaxes controlled by soil moisture, soil nutrients, topography, slope exposure, fire, and animal activity. Climax pattern theory was proposed by Whittaker 1953. The climax pattern theory recognizes a variety of climaxes governed by responses of species populations to biotic and abiotic conditions. According to this theory, the total environment of the ecosystem determines the composition, species structure, and balance of a climax community. The environment includes the species' responses to moisture, temperature, and nutrients, their biotic relationships, availability of flora and fauna to colonize the area, chance dispersal of seeds and animals, soils, climate, and disturbance such as fire and wind. The nature of climax vegetation will change as the environment changes. The climax community represents a pattern of populations that corresponds to and changes with the pattern of environment. The central and most widespread community is the climatic climax. The theory of alternative stable states suggests there is not one endpoint but many which transition between each other over ecological time. Succession by habitat type. Forest succession. Forests, being an ecological system, are subject to the species succession process. 36. There are opportunistic, or pioneer, species that produce great quantities of seed that are disseminated by the wind, and therefore can colonize big empty extensions. 37. They are capable of germinating and growing in direct sunlight. Once they have produced a closed canopy, the lack of direct sun radiation at the soil makes it difficult for their own seedlings to develop. It is then the opportunity for shade-tolerant species to become established under the protection of the pioneers. When the pioneers die, the shade-tolerant species replace them. These species are capable of growing beneath the canopy, and therefore, in the absence of disturbances, will stay. For this reason it is then said the stand has reached its climax. When a disturbance occurs, the opportunity for the pioneers opens up again, provided they are present or within a reasonable range. An example of pioneer species, in forests of northeastern North America are Betula papyrifera, white birch, and Prunus serratina, black cherry, that are particularly well adapted to exploit large gaps in forest canopies, but are intolerant of shade and are eventually replaced by other shade-tolerant species in the absence of disturbances that create such gaps. In the tropics, well-known pioneer forest species can be found among the genera Cecropia, Acroma and Trema. 37. Things in nature are not black and white, and there are intermediate stages. It is therefore normal that between the two extremes of light and shade there is a gradient, and there are species that may act as pioneer or tolerant, depending on the circumstances. It is of paramount importance to know the tolerance of species in order to practice an effective silviculture. Wetland succession. Since many types of wetland environments exist, succession may follow a wide array of trajectories and patterns in wetlands. Under the classical model, the process of secondary succession holds that a wetland progresses over time from an initial state of open water with few plants, to a forested climax state where decayed organic matter has built up over time, forming peat. However, many wetlands are maintained by regular disturbance or natural processes at an equilibrium state that does not resemble the predicted forested climax. 38. The idea that ponds and wetlands gradually fill in to become dry land has been criticized and called into question due to lack of evidence. 5. Wetland succession is a uniquely complex, nonlinear process shaped by hydrology. 39. Hydrological factors often work against linear processes that predict a succession to a climax state. The energy carried by moving water may create a continuous source of disturbance. For example, in coastal wetlands, the tides moving in and out continuously acts upon the ecological community. Fire may also maintain an equilibrium state in a wetland by burning off vegetation, thus interrupting the accumulation of peat. 38. Water entering and leaving the wetland follows patterns that are broadly cyclical but erratic. For example, seasonal flooding and drying may occur with yearly changes in precipitation, causing seasonal changes in the wetland community that maintain it at a stable state. 5. However, unusually heavy rain or unusually severe drought may cause the wetland to enter a positive feedback loop where it begins to change in a linear direction. 39. Since wetlands are sensitive to changes in the natural processes that maintain them, human activities, invasive species, and climate change could initiate long-term changes in wetland ecosystems. 38. Grassland succession. For a long time, grasslands were thought to be early stages of succession, dominated by weedy species and with little conservation value. However, comparing grasslands that form after recovery from long-term disruptions like agricultural tillage with ancient or old growth grasslands has Ecology portal, Biology portal, Environment portal shown that grasslands are not inherently early successional communities. Rather, grasslands undergo a centuries-long process of succession, and a grassland that is tilled up for agriculture or otherwise destroyed is estimated to take a minimum of 100 years, and potentially on average 1,400 years, to recover to its previous level of biodiversity. 40. However, planting a high diversity of late successional grassland species in a disturbed environment can accelerate the recovery of the soil's ability to sequester carbon, resulting in twice as much carbon storage as a naturally recovering grassland over the same period of time. 41. Many grassland ecosystems are maintained by disturbance, such as fire and grazing by large animals, or else the process of succession will change them to forest or shrubland. In fact, it is debated whether fire should be considered disturbance at all for the North American prairie ecosystems, since it maintains, rather than disrupts, an equilibrium state. 42. Many late successional grassland species have adaptations that allow them to store nutrients underground and resprout rapidly after, above ground, disturbances like fire or grazing. Disturbance events that severely disrupt or destroy the soil, such as tilling, eliminate these late successional species, reverting the grassland to an early successional stage dominated by pioneers, whereas fire and grazing benefit late successional species. 40. Both too much and too little disturbance can damage the biodiversity of disturbance-dependent ecosystems like grasslands. 43. In North American semi-arid grasslands, the introduction of livestock ranching and absence of fire was observed to cause a transition away from grasses to woody vegetation, particularly mesquite. 44. However, the means by which ecological succession under frequent disturbance results in ecosystems of the sort seen in remnant prairies is poorly understood. 45. 43. Species richness. Species richness is the number of different species represented in an ecological community, landscape or region. 1. Species richness is simply a count of species, and it does not take into account the abundances of a species or their relative abundance distributions. Species richness is sometimes considered synonymous with species diversity, but the formal metric species diversity takes into account both species richness and species evenness. Species richness has proven to be a positive representation to show how species interaction in ecosystems can lead to the productivity and growth of biodiversity. 2. Global mammal richness, 2015. Habitual interactions among aquatic species. Biodiversity is an imperative factor used to connect both species richness and species evenness to detect relatedness where genetics, relative species abundances and ecological distributions is concerned. The higher biodiversity there is within an ecosystem, the higher the chances are that species richness will be prevalent with respects to the ecosystem's relative abundance levels. Species richness across different parts of the world will show variations based on location, climate, predator-prey relationship, food availability and other factors that lies in the throes of environmental influences. The image below shows a representation of an aquatic relationship among members of the same community. Competition for food, mating spaces, and overall predator or prey relationship can also arise. An abundance in the number of species will present itself where habitats are relatively available for species to live, where competition and predators are not actively seeking to lower their abundance levels. 3. The connection between species richness and diversity. Biodiversity pyramid. As previously mentioned, biological diversity, also known as biodiversity, is the variation living things and how their interactions with each other affect life on Earth. With biodiversity, there are few things that contribute to the life on Earth. One of those things are species such as new species being discovered or species becoming extinct. Since species richness represents the number of species living in space, it plays a part in not only biodiversity but with species diversity as well. Knowing the number of species in a space is very essential when looking on the different living things that contribute to the diversity of either Earth or within a certain living space, because we can observe the changes that is constantly occurring throughout life. When using species richness to compare the diversity, there are a few factors that needs to be considered. Those factors include looking at the overall and the relative abundance of the individual within the species group and where the species in the living space are mostly found. For without those factors, it would be hard to determine the diversity of species. Sampling considerations. Depending on the purposes of quantifying species richness, the individuals can be selected in different ways. They can be, for example, trees found in an inventory plot, birds observed from a monitoring point, or beetles collected in a pitfall trap. Once the set of individuals has been defined, its species richness can be exactly quantified, provided the species-level taxonomy of the organisms of interest is well enough known. Applying different species delimitations will lead to different species richness values for the same set of individuals. In practice, people are usually interested in the species richness of areas so large that not all individuals in them can be observed and identified to species. Then applying different sampling methods will lead to different sets of individuals being observed for the same area of interest, and the species richness of each set may be different. When a new individual is added to a set, it may introduce a species that was not yet represented in the set, and thereby increase the species richness of the set. For this reason, sets with many individuals can be expected to contain more species than sets with fewer individuals. If species richness of the obtained sample is taken to represent species richness of the underlying habitat or other larger unit, values are only comparable if sampling efforts are standardized in an appropriate way. Resampling methods can be used to bring samples of different sizes to a common footing. 5 properties of the sample, especially the number of species only represented by one or a few individuals, can be used to help estimating the species richness in the population from which a sample was drawn. 6, 7, 8 These indications and adaptations to the detail of population enhancements can provide further diversity in urban ecological areas by means of promoting species richness while also considering the ecological community. Trends in species richness. The observed species richness is affected not only by the number of individuals but also by the heterogeneity of the sample. If individuals are drawn from different environmental conditions or different habitats, the species richness of the resulting set can be expected to be higher than if all individuals are drawn from similar environments. The accumulation of new species with increasing sampling effort can be visualized with a species accumulation curve. If the observed species richness is affected not only by the number of individuals but also by the heterogeneity of the sample. If individuals are drawn from different environmental conditions or different habitats, the species richness of the resulting set can be expected to be higher than if all individuals are drawn from similar environments. The accumulation of new species with increasing sampling effort can be visualized with a species accumulation curve. Such curves can be constructed in different ways. 9. Increasing the area sampled increases observed species richness both because more individuals get included in the sample and because large areas are environmentally more heterogeneous than small areas. The higher biodiversity there is within an ecosystem, the higher the chances are that species richness will be prevalent with respects to the ecosystem's relative abundance levels. Species richness across different parts of the world will show variations based on location, climate, predator-prey relationship, food availability and other factors that lies in the throes of environmental influences. The image below shows a representation of an aquatic relationship among members of the same community. Competition for food, mating spaces, and overall predator or prey relationship can also arise. An abundance in the number of species will present itself where habitats are relatively available for species to live, where competition and predators are not actively seeking to lower their abundance levels. 3. The connection between species richness and diversity. Biodiversity pyramid. As previously mentioned, biological diversity, also known as biodiversity, is the variation living things and how their interactions with each other affect life on Earth. With biodiversity, there are few things that contribute to the life on Earth. One of those things are species, such as new species being discovered or species becoming extinct. Since species richness represents the number of species living in space, it plays a part in not only biodiversity but with species diversity as well. Knowing the number of species in a space is very essential when looking on the different living things that contribute to the diversity of either Earth or within a certain living space, because we can observe the changes that is constantly occurring throughout life. When using species richness to compare the diversity, there are few factors that needs to be considered. Those factors include looking at the overall and the relative abundance of the individual within the species group and where the species in the living space are mostly found. For without those factors, it would be hard to determine the diversity of species. Sampling considerations. Depending on the purposes of quantifying species richness, the individuals can be selected in different ways. They can be, for example, trees found in an inventory plot, birds observed from a monitoring point, or beetles collected in a pitfall trap. Once the set of individuals has been defined, its species richness can be exactly quantified, provided the species-level taxonomy of the organisms of interest is well enough known. Applying different species delimitations will lead to different species richness values for the same set of individuals. In practice, people are usually interested in the species richness of areas so large that not all individuals in them can be observed and identified to species. Then applying different sampling methods will lead to different sets of individuals being observed for the same area of interest, and the species richness of each set may be different. When a new individual is added to a set, it may introduce a species that was not yet represented in the set, and thereby increase the species richness of the set. For this reason, sets with many individuals can be expected to contain more species than sets with fewer individuals. If species richness of the obtained sample is taken to represent species richness of the underlying habitat or other larger unit, values are only comparable if sampling efforts are standardized in an appropriate way. Resampling methods can be used to bring samples of different sizes to a common footing. 5 properties of the sample, especially the number of species only represented by one or a few individuals, can be used to help estimating the species richness in the population from which a sample was drawn. 6, 7, 8 These indications and adaptations to the detail of population enhancements can provide further diversity in urban ecological areas by means of promoting species richness while also considering the ecological community. Trends in species richness. The observed species richness is affected not only by the number of individuals but also by the heterogeneity of the sample. If individuals are drawn from different environmental conditions or different habitats, the species richness of the resulting set can be expected to be higher than if all individuals are drawn from similar environments. The accumulation of new species with increasing sampling effort can be visualized with a species accumulation curve. Such curves can be constructed in different ways. 9. Increasing the area sampled increases observed species richness both because more individuals get included in the sample and because large areas are environmentally more heterogeneous than small areas. Many organism groups have most species in the tropics, which leads to latitudinal gradients in species richness. There has been much discussion about the relationship between productivity and species richness. Results have varied among studies, such that no global consensus on either the pattern or its possible causes has emerged. 10. Species diversity and richness is likely to occur in areas of warmer climates because of variability in food types, mating opportunities, urban area provision of cleaner environments and other factors that can lead to an improved species richness. 11. Species richness also depicts immense extension by means of expanding in terms of ecological and environmental availability of urban food types, which enriches the likelihood of species richness increasing even in urban areas. Additionally, increasing food availability in certain environments will provide better chances of species richness in terms of diversity. 12. Where there is food, there are animals according to the balance of nature or the ecological balance as it is generally known. 13. Which expresses types of food chain and food webs which are both used in urban ecology to show the differences but mostly similarities in which feeding relations between species can be understood. 14. Every type of species will consist of their individual type of feeding relationship with organisms provided by the environment. 15. However, it is necessary to understand that feeding relationships in the urban species will indeed form a type of symbiotic relationship within an ecosystem according to urban necessities as shown in the image provided. 16. Feeding relationship. Applications. Species richness is often used as a criterion when assessing the relative conservation values of habitats or landscapes. However, species richness is blind to the identity of the species. An area with many endemic or rare species is generally considered to have higher conservation value than another area where species richness is similar, but all the species are common and widespread. Location-wise, urban settings can influence species richness by means of proper environmental conservation, availability of safety and other factors like water, trees, and sustainable habitat. Species thrives in areas where they are provided with the basic necessities for their survival. Even in urban areas where they may face challenges like competition or predators, they still have a high chance of surviving when placed in an environment that provides adequate resources that can benefit the species and promote species diversity and richness. 17. With food web in mind, species richness in either aquatic or non-aquatic environments can serve as either predator or prey for some animals. In this case, it is possible for species richness with respect to species evenness to form a negative outcome when the relative species abundance is threatened. There can also be a negative or positive impact on species diversity. 18. This can also influence how species richness in an area will affect their environment. Since some environments thrives off of species interactions, it can pose an undesired consequence whereby the community itself is impacted by the loss of species. Species richness and urban ecology. Urban ecology and species richness commonly coincides based on the location, availability of resources, suitable temperature, access to water and other factors like typical wildlife of all sorts. The richness and abundance of species are the starting elements that promotes the growth and survival of urban ecological life. Without the community's input, it may be difficult to accurately represent an urban life if certain accommodations are not met. Urban ecology always starts as an interactive relationship between species and surroundings to show an interconnected type of relationship between species richness and the response of the environment. 19. Global urban development has been established to increase the likelihood of both humans and different types of species living together in harmony not just for the benefit of the world, but for each individual ecosystem. When humans and animals work together, species diversity can benefit whereby animals can freely exist in their own world without the fear of being slaughtered just for coexisting with humans, and humans can also benefit from the help of animals. It is not always impossible for humans and animals to exist in the same space. With the help of humans, species can be able to live in peaceful neighborhoods or habitats within their own biodiversity. All this does is increase the likelihood of species richness and an abundance of population growth. 20. Additionally, with the concept of species richness, species abundance, population growth and diversity in mind, it is crucial to understand that urban ecology has not always been stable when change is constant. Environmental and ecological resilience has played major roles in the redevelopment of habitats, ecosystems, biomes and their impacts and adjustments to urbanization. 21. Change is a necessary factor where species richness is concerned and attached to the environmental and ecological growth and development across the globe. Change is inevitable, and for the sake of species richness, it's necessary that they are all exposed to places and communities they can have positive connections with, not only for the benefit of the now, but for their future generations. An example of an urbanization area. Species richness can be affected by urbanization. Since urbanization involves improve areas to make more modern, that means that the areas will undergo changes to better life for some humans, but the other living species such as animals will not get the same benefits as humans. When people think of urbanization, they don't think about how it affects the species in the soil. It can be observed that species richness within the soil will decrease as urbanization develops due to the environment changes. For instance, an experiment was conducted to see how the soil species with the high ability and low ability to move throughout the soil were affected due to the changes that came with urbanization. The result was that the species that had low ability of movement and medium ability of movement such as snails and annelids was affected by urbanization drying out the soil, which limited the species' movement. The drying of the soil causes those species mentioned to find a new space with soil that is moist to help move through the soil. 22. In conclusion, species richness is affected by how urban the area is. The more urban, the less the richness of the species depending on if the species are adjusted to new changes in their home as there and vice versa. Species evenness. Species evenness describes the commonness or rarity of a species. It requires knowing the abundance of each species relative to those of the other species within the community. 1. Abundance values can be difficult to obtain. Area-based counts, distance methods, and mark recapture studies are the three general categories of methods for estimating abundance. Species evenness is combined with species richness, the number of species in the community, in order to determine species diversity, which is an important measure of community structure. Community structure in turn provides the quantitative basis needed to create hypotheses and experiments that help to increase understanding of how communities work. To demonstrate the contributions of species richness and species evenness to species diversity, consider the following hypothetical example, in which there are two meadow communities, each containing 4 species of butterflies. Hence, both communities have the same butterfly species richness, however their species evenness differs. In Community A, one of the species constitutes 80% of the individuals in the community, while the remaining 3 species comprise only 20% of the individuals in the community. In Community B, the number of individuals are evenly divided among the 4 species, 25% each. Therefore, Community A has lower species evenness than Community B when comparing the two communities. Even though each has the same species richness of 4 species, Community B has a higher species diversity because it has higher species evenness. The Shannon index is the most commonly used way to quantitatively determine species diversity, H, as modeled by the following equation. The Shannon index factors in both species evenness and species richness, as represented by the variables pi and s, respectively. The lowest possible value of H is 0, and the higher a community's H value, the greater its species diversity. If calculated for each of the 2 hypothetical butterfly communities described previously, the lower Shannon index value H would belong to Community A, thereby confirming mathematically that this community has lower species diversity than Community B. Furthermore, given that both communities have the same species richness, it is the lower species evenness in Community A that drives the difference in species diversity. Species diversity. Species diversity is the number of different species that are represented in a given community, a dataset. The effective number of species refers to the number of equally abundant species needed to obtain the same mean proportional species abundance as that observed in the dataset of interest, where all species may not be equally abundant. Meanings of species diversity may include species richness, taxonomic or phylogenetic diversity, and/or species evenness. Species richness is a simple count of species. Taxonomic or phylogenetic diversity is the genetic relationship between different groups of species. Species evenness quantifies how equal the abundance is of the species. 1, 2, 3. Calculation of diversity. Species diversity in a dataset can be calculated by first taking the weighted average of species proportional abundances in the dataset, and then taking the inverse of this. The equation is 1, 2, 3. Species diversity. Species diversity is the number of different species that are represented in a given community, a dataset. The effective number of species refers to the number of equally abundant species needed to obtain the same mean proportional species abundance as that observed in the dataset of interest, where all species may not be equally abundant. Meanings of species diversity may include species richness, taxonomic or phylogenetic diversity, and/or species evenness. Species richness is a simple count of species. Taxonomic or phylogenetic diversity is the genetic relationship between different groups of species. Species evenness quantifies how equal the abundance is of the species. 1, 2, 3. Calculation of diversity. Species diversity in a dataset can be calculated by first taking the weighted average of species proportional abundances in the dataset, and then taking the inverse of this. The equation is 1, 2, 3. The denominator equals mean proportional species abundance in the dataset is calculated with the weighted generalized mean with exponent q1. In the equation, S is the total number of species richness, in the dataset, and the proportional abundance of the ith species is. The proportional abundances themselves are used as weights. The equation is often written in the equivalent form. The value of q determines which mean is used. Q equals 0 corresponds to the weighted harmonic mean, which is 1s because the values cancel out, with the result that 0d is equal to the number of species or species richness. SQ equals 1 is undefined, except that the limit as q approaches 1 is well defined, 4. Which is the exponential of the Shannon entropy. Q equals 2 corresponds to the arithmetic mean. As q approaches infinity, the generalized mean approaches the maximum value. In practice, q modifies species weighting, such that increasing q increases the weight given to the most abundant species, and fewer equally abundant species are hence needed to reach mean proportional abundance. Consequently, large values of q lead to smaller species diversity than small values of q for the same dataset. If all species are equally abundant in the dataset, changing the value of q has no effect, but species diversity at any value of q equals species richness. Negative values of q are not used, because then the effective number of species diversity would exceed the actual number of species richness. As q approaches negative infinity, the generalized mean approaches the minimum value. In many real datasets, the least abundant species is represented by a single individual, and then the effective number of species would equal the number of individuals in the dataset. 2, 3. The same equation can be used to calculate the diversity in relation to any classification, not only species. If the individuals are classified into genera or functional types, represents the proportional abundance of the ith genus or functional type, and qd equals genus diversity or functional type diversity, respectively. Diversity indices. Often researchers have used the values given by one or more diversity indices to quantify species diversity. Such indices include species richness, the Shannon index, the Simpson index, and the complement of the Simpson index, also known as the Gini-Simpson index. 5, 6, 7. When interpreted in ecological terms, each one of these indices corresponds to a different thing, and their values are therefore not directly comparable. Species richness quantifies the actual rather than effective number of species. The Shannon index equals log 1d, that is, q approaching 1, and in practice quantifies the uncertainty in the species identity of an individual that is taken at random from the dataset. The Simpson index equals 1/2 d, q equals 2, and quantifies the probability that 2 individuals taken at random from the dataset with replacement of the first individual before taking the second represent the same species. The Gini-Simpson index equals 1.5D and quantifies the probability that the two randomly taken individuals represent different species. 1, 2, 3, 7, 8. Sampling considerations. Depending on the purposes of quantifying species diversity, the dataset used for the calculations can be obtained in different ways. Although species diversity can be calculated for any dataset. Environment Portal. Ecology Portal. Earth Sciences Portal. Where individuals have been identified to species, meaningful ecological interpretations require that the dataset is appropriate for the questions at hand. In practice, the interest is usually in the species diversity of areas so large that not all individuals in them can be observed and identified to species, but a sample of the relevant individuals has to be obtained. Extrapolation from the sample to the underlying population of interest is not straightforward, because the species diversity of the available sample generally gives an underestimation of the species diversity in the entire population. Applying different sampling methods will lead to different sets of individuals being observed for the same area of interest, and the species diversity of each set may be different. When a new individual is added to a dataset, it may introduce a species that was not yet represented. How much this increases species diversity depends on the value of Q. When Q equals 0, each new actual species causes species diversity to increase by one effective species, but when Q is large, adding a rare species to a dataset has little effect on its species diversity. 9. In general, sets with many individuals can be expected to have higher species diversity than sets with fewer individuals. When species diversity values are compared among sets, sampling efforts need to be standardized in an appropriate way for the comparisons to yield ecologically meaningful results. Resampling methods can be used to bring samples of different sizes to a common footing. 10, 11. Species discovery curves and the number of species only represented by one or a few individuals can be used to help in estimating how representative the available sample is of the population from which it was drawn. 12, 13. The observed species diversity is affected not only by the number of individuals but also by the heterogeneity of the sample. If individuals are drawn from different environmental conditions or different habitats, the species diversity of the resulting set can be expected to be higher than if all individuals are drawn from a similar environment. Increasing the area sampled increases observed species diversity both because more individuals get included in the sample and because large areas are environmentally more heterogeneous than small areas. Alpha diversity, beta diversity. Diversity index. A diversity index is a method of measuring how many different types, e.g. species, there are in a dataset, e.g. a community. Some more sophisticated indices also account for the phylogenetic relatedness among the types. 1. Diversity indices are statistical representations of different aspects of biodiversity, e.g. richness, evenness, and dominance, which are useful simplifications for comparing different communities or sites. Effective number of species are Hill numbers. When diversity indices are used in ecology, the types of interest are usually species, but they can also be other categories, such as genera, families, functional types, or haplotypes. The entities of interest are usually individual organisms, e.g. plants or animals, and the measure of abundance can be, for example, number of individuals, biomass or coverage. In demography, the entities of interest can be people, and the types of interest various demographic groups. In information science, the entities can be characters and the types of the different letters of the alphabet. The most commonly used diversity indices are simple transformations of the effective number of types, also known as true diversity, but each diversity index can also be interpreted in its own right as a measure corresponding to some real phenomenon, but a different one for each diversity. Index 2, 3, 4, 5. Many indices only account for categorical diversity between subjects or entities. Such indices, however, do not account for the total variation diversity that can be held between subjects or entities, which occurs only when both categorical and qualitative diversity are calculated. True diversity, or the effective number of types, refers to the number of equally abundant types needed for the average proportional abundance of the types to equal that observed in the dataset of interest, where all types may not be equally abundant. The true diversity in a dataset is calculated by first taking the weighted generalized mean MQ-1 of the proportional abundances of the types in the dataset, and then taking the reciprocal of this. The equation is 4, 5. The denominator MQ-1 equals the average proportional abundance of the types in the dataset is calculated with the weighted generalized mean with exponent Q-1. In the equation, R is richness, the total number of types in the dataset, and the proportional abundance of the ith type is pi. The proportional abundances themselves are used as the nominal weights. The numbers are called Hill numbers of order Q, or effective number of species. 6. When Q equals 1, the above equation is undefined. However, the mathematical limit as Q approaches 1 is well defined, and the corresponding diversity is calculated with the following equation, which is the exponential of the Shannon entropy calculated with natural logarithms, see above. In other domains, this statistic is also known as the perplexity. The general equation of diversity is often written in the form 2, 3. And the term inside the parentheses is called the basic sum. Some popular diversity indices correspond to the basic sum as calculated with different values of Q. 3. Sensitivity of the diversity value to rare versus abundant species. The value of Q is often referred to as the order of the diversity. It defines the sensitivity of the true diversity to rare versus abundant species by modifying how the weighted mean of the species' proportional abundances is calculated. With some values of the parameter Q, the value of the generalized mean MQ-1 assumes familiar kinds of weighted means as special cases. In particular, Q equals 0 corresponds to the weighted harmonic mean, Q equals 1 to the weighted geometric mean, and Q equals 2 to the weighted arithmetic mean. As q approaches infinity, the weighted generalized mean with exponent q-1 approaches the maximum pi value, which is the proportional abundance of the most abundant species in the dataset. Generally, increasing the value of q increases the effective weight given to the most abundant species. This leads to obtaining a larger mq-1 value and a smaller true diversity qd value with increasing q. When q equals 1, the weighted geometric mean of the pi values is used, and each species is exactly weighted by its proportional abundance. In the weighted geometric mean, the weights are the exponents. When q greater than 1, the weight given to abundant species is exaggerated, and when q less than 1, the weight given to rare species is. At q equals 0, the species weights exactly cancel out the species' proportional abundances, such that the weighted mean of the pi values equals 1 r even when all species are not equally abundant. At q equals 0, the effective number of species, 0d, hence equals the actual number of species r. In the context of diversity, q is generally limited to non-negative values. This is because negative values of q would give rare species so much more weight than abundant ones that d would exceed r. 4. 5. Richness. Richness r simply quantifies how many different types the dataset of interest contains. For example, species richness, usually noted S, is simply the number of species, e.g. at a particular site. Richness is a simple measure, so it has been a popular diversity index in ecology, where abundance data are often not available. 7. If true diversity is calculated with q equals 0, the effective number of types, 0d, equals the actual number of types, which is identical to richness r. 3. 5. Shannon Index. The Shannon Index has been a popular diversity index in the ecological literature, where it is also known as Shannon's Diversity Index, Shannon-Wiener Index, and, erroneously, Shannon-Weaver Index. 8. The measure was originally proposed by Claude Shannon in 1948 to quantify the entropy, hence Shannon entropy, related to Shannon information content in strings of text. 9. The idea is that the more letters there are, and the closer the proportional abundance is in the string of interest, the more difficult it is to correctly predict which letter will be the next one in the string. The Shannon entropy quantifies the uncertainty, entropy or degree of surprise, associated with this prediction. It is most often calculated as follows. Where pi is the proportion of characters belonging to the ith type of letter in the string of interest. In ecology, pi is often the proportion of individuals belonging to the ith species in the dataset of interest. Then the Shannon entropy quantifies the uncertainty in predicting the species identity of an individual that is taken at random from the dataset. Although the equation is here written with natural logarithms, the base of the logarithm used when calculating the Shannon entropy can be chosen freely. Shannon himself discussed logarithm bases 2, 10 and e, and these have since become the most popular bases in applications that use the Shannon entropy. Each log base corresponds to a different measurement unit, which has been called binary digits, bits, decimal digits, decits, and natural digits, nats, for the bases 2, 10 and e, respectively. Comparing Shannon entropy values that were originally calculated with different log bases requires converting them to the same log base. Change from the base A to base B is obtained with multiplication by log BA. 9. The Shannon index H is related to the weighted geometric mean of the proportional abundances of the types. Specifically, it equals the logarithm of true diversity as calculated with q equals 1, 4. This can also be written which equals. Since the sum of the pi values equals 1 by definition, the denominator equals the weighted geometric mean of the pi. Since the sum of the pi values equals 1 by definition, the denominator equals the weighted geometric mean of the pi values, with the pi values themselves being used as the weights exponents in the equation. The term within the parentheses hence equals true diversity 1d, and h equals lane 1d. 2, 4, 5. When all types in the dataset of interest are equally common, all pi values equal 1 r, and the Shannon index hence takes the value lane r. The more unequal the abundances of the types, the larger the weighted geometric mean of the pi values, and the smaller the corresponding Shannon entropy. If practically all abundance is concentrated to one type, and the other types are very rare even if there are many of them, Shannon entropy approaches 0. When there is only one type in the dataset, Shannon entropy exactly equals 0, there is no uncertainty in predicting the type of the next randomly chosen entity. In machine learning, the Shannon index is also called as information gain. Rainy entropy. The Rainy entropy is a generalization of the Shannon entropy to other values of q than 1. It can be expressed which equals. This means that taking the logarithm of true diversity based on any value of q gives the Rainy entropy corresponding to the same value of q. Simpson index. The Simpson index was introduced in 1949 by Edward H. Simpson to measure the degree of concentration when individuals are classified into types. 10. The same index was rediscovered by Oris C. Herfindahl in 1950. 11. The square root of the index had already been introduced in 1945 by the economist Albert O. Hirschman. 12. As a result, the same measure is usually known as the Simpson index in ecology, and as the Herfindahl index or the Herfindahl-Hirschman index (HHI) in economics. The measure equals the probability that two entities taken at random from the dataset of interest represent the same type. 10. It equals. Where r is richness, the total number of types in the dataset. This equation is also equal to the weighted arithmetic mean of the proportional abundances pi of the types of interest, with the proportional abundances themselves being used as the weights. 2. Proportional abundances are by definition constrained to values between 0 and 1, but it is a weighted arithmetic mean, hence lambda is greater than or equal to 1 r, which is reached when all types are equally abundant. By comparing the equation used to calculate lambda with the equations used to calculate true diversity, it can be seen that 1 lambda equals 2d, i.e., true diversity is calculated with q equals 2. The original Simpson's index hence equals the corresponding basic sum. 3. The interpretation of lambda as the probability that two entities taken at random from the dataset of interest represent the same type assumes that the first entity is replaced to the dataset before taking the second entity. If the dataset is very large, sampling without replacement gives approximately the same result, but in small datasets, the difference can be substantial. If the dataset is small, and sampling without replacement is assumed, the probability of obtaining the same type with both random draws is where n is the number of entities belonging to the ith type and n is the total number of entities in the dataset. 10. This form of the Simpson index is also known as the Hunter-Gaston index in microbiology. 13. Since the mean proportional abundance of the types increases with decreasing number of types and increasing abundance of the most abundant type, lambda obtains small values in datasets of high diversity and large values in datasets of low diversity. This is counterintuitive behavior for a diversity index, so often, such transformations of lambda that increase with increasing diversity have been used instead. The most popular of such indices have been the inverse Simpson index, 1 lambda, and the Gini-Simpson index, 1 minus lambda. 2, 3. Both of these have also been called the Simpson index in the ecological literature, so care is needed to avoid accidentally comparing the different indices as if they were the same. Inverse Simpson index. The inverse Simpson index equals. This simply equals true diversity of order 2, i.e. the effective number of types that is obtained when the weighted arithmetic mean is used to quantify average proportional abundance of types in the dataset of interest. The index is also used as a measure of the effective number of parties. Gini-Simpson index. The Gini-Simpson index is also called Gini impurity, or Gini's diversity index 14 in the field of machine learning. The original Simpson index lambda equals the probability that two entities taken at random from the dataset of interest with replacement represent the same type. Its transformation 1 minus lambda, therefore, equals the probability that the two entities represent different types. This measure is also known in ecology as the probability of interspecific encounter, pi 15, and the Gini-Simpson index. 3. It can be expressed as a transformation of the true diversity of order 2, the Gibbs-Martin index of sociology, psychology, and management studies, 16, which is also known as the Blau index, is the same measure as the Gini-Simpson index. The quantity is also known as the expected heterozygosity in population genetics. Berger-Parker index. The Berger-Parker index, named after Wolfgang H. Berger and Francis Lawrence Parker, 17, equals the maximum pi value in the dataset, i.e., the proportional abundance of the most abundant type. This corresponds to the weighted generalized mean of the pi values when q approaches infinity, and hence equals the inverse of the true diversity of order infinity, 1, infinity d. See species area relationship. The species area relationship or species area curve describes the relationship between the area of a habitat, or of part of a habitat, and the number of species found within that area. Larger areas tend to contain larger numbers of species, and empirically, the relative numbers seem to follow systematic mathematical relationships. 1. The species area relationship is usually constructed for a single type of organism, such as all vascular plants or all species of a specific trophic level within a particular site. It is rarely, if ever, constructed for all types of organisms if simply because of the prodigious data requirements. It is related but not identical to the species discovery curve. The species area relationship for a contiguous habitat. Ecologists have proposed a wide range of factors determining the slope and elevation of the species area relationship. 2. These factors include the relative balance between immigration and extinction. 3. Rate and magnitude of disturbance on small versus large areas. 3. Predator-prey dynamics. 4. And clustering of individuals of the same species as a result of dispersal limitation or habitat heterogeneity. 5. The species area relationship has been reputed to follow from the second law of thermodynamics. 6. In contrast to these mechanistic explanations, others assert the need to test whether the pattern is simply the result of a random sampling process. 7. Species area relationships are often evaluated in conservation science in order to predict extinction rates in the case of habitat loss and habitat fragmentation. 8. Authors have classified the species area relationship according to the type of habitats being sampled and the census design used. Frank W. Preston, an early investigator of the theory of the species area relationship, divided it into two types: samples, a census of a contiguous habitat that grows in the census area, also called mainland, species area relationships, and isolates, a census of discontiguous habitats, such as islands, also called island, species area relationships. 1. Michael Rosenzweig also notes that species area relationships for very large areas, those collecting different biogeographic provinces or continents, behave differently from species area relationships from islands or smaller contiguous areas. 2. It has been presumed that island-like species area relationships have steeper slopes in log-log space than mainland relationships. 2. But a 2006 meta-analysis of almost 700 species area relationships found the former had lower slopes than the latter. 9. Regardless of census design and habitat type, species area relationships are often fitted with a simple function. Frank Preston advocated the power function based on his investigation of the log-normal species abundance distribution. 1. If is the number of species, is the habitat area, and is the slope of the species area relationship in log-log space, then the power function species area relationship goes as Here is a constant which depends on the unit used for area measurement, and equals the number of species that would exist if the habitat area was confined to 1 square unit. The graph looks like a straight line on log-log axis, and can be linearized as. In contrast, Henry Gleason championed the semilog model, which looks like a straight line on semilog axis, where the area is logged and the number of species is arithmetic. In either case, the species-area relationship is almost always decelerating, has a negative second derivative when plotted arithmetically. 10. Species-area relationships are often graphed for islands or habitats that are otherwise isolated from one another, such as woodlots in an agricultural landscape of different sizes. 3. Although larger islands tend to have more species, a smaller island may have more than a larger one. In contrast, species-area relationships for contiguous habitats will always rise as areas increases, provided that the sample plots are nested within one another. The species-area relationship for mainland areas, contiguous habitats, will differ according to the census design used to construct it. 11. A common method is to use quadrats of successively larger size so that the area enclosed by each one includes the area enclosed by the smaller one, i.e. areas are nested. In the first part of the 20th century, plant ecologists often use the species area curve to estimate the minimum size of a quadrat necessary to adequately characterize a community. This is done by plotting the curve usually on arithmetic axes, not log-log or semilog axes, and estimating the area after which using larger quadrats results in the addition of only a few more species. This is called the minimal area. A quadrat that encloses a minimal area is called a releve, and using species area curves in this way is called the releve method. It was largely developed by the Swiss ecologist Josias Braun-Blanquet. 12. Estimation of the minimal area from the curve is necessarily subjective, so some authors prefer to define the minimal area as the area enclosing at least 95% or some other large proportion of the total species found. The problem with this is that the species area curve does not usually approach an asymptote, so it is not obvious what should be taken as the total. 12. The number of species always increases with area up to the point where the area of the entire world has been accumulated. 13. See also. The theory of island biogeography. The Theory of Island Biogeography is a 1967 book by the ecologist Robert MacArthur and the biologist Edward O. Wilson. 1. It is widely regarded as a seminal work in island biogeography and ecology. The Princeton University Press reprinted the book in 2001 as a part of the Princeton Landmarks in Biology series. 1. The book popularized the theory that insular biota maintain a dynamic equilibrium between immigration and extinction rates. The book also popularized the concepts and terminology of RK selection theory. 2. Background. The theory of island biogeography has its roots in Wilson's work on the ants of Melanesia. MacArthur synthesized Wilson's ideas about competition, colonization and equilibrium into a simple graphical representation of immigration and extinction curves, from which one can determine the equilibrial species number on an island. 3. MacArthur and Wilson's ideas were first presented in a paper published in 1963, 4. and were further developed into a book. 3. Summary. Introduction. In the introductory chapter, MacArthur and Wilson highlight the significance of studying island biogeography. Since islands are less complex and more numerous than larger ecosystems, islands provide better opportunities to develop insights and perform replicable field research. Given that insular microcosms are common to all ecosystems, principles from island biogeography can be applied generally. This graph portrays the immigration rate of new species and the extinction rate of resident species versus the number of species present on an island. The intersection point gives the equilibrial species number. Area diversity pattern. In chapters 2 and 3, MacArthur and Wilson postulate that insular species richness depends on island size and isolation from source regions. The authors present an equilibrium model that is based on the following concept: when there is an addition of the number of species on an island, the island's immigration rate of new species will decrease while the extinction rate of resident species will increase. MacArthur and Wilson thus assume that there will be an equilibrial point where the immigration rate equals the extinction rate. They further hypothesize that an increase in island size will lower extinction curves while a decrease in distance between the island and the source region will raise immigration curves. Since the intersection of immigration and extinction rate curves determines the species number, the authors predict that larger islands will have more species than smaller islands, assuming these islands are comparably isolated, and isolated islands will have fewer species than islands more proximal to source regions, assuming these islands are equally large. There is additional discourse on how insular clusters and stepping stones affect this model. Modeling colonization and dispersion. Chapter 4 discusses survivorship theory. The authors describe a model which states that the probability for successful colonization is dependent on birth rate, death rate, and carrying capacity of the environment. From this model, conclusions are made on the average survival time for a propagule's offspring, the average survival time of a saturated population, and characteristics of successful propagules. In Chapter 5, MacArthur and Wilson examine why species can be excluded from insular environments and how the niche of a species changes after introduction. The authors surmise that pioneering species can be excluded for the following reasons. The island has saturated levels of pre-existing competition, the pioneering species cannot maintain a population large enough to avoid extinction, and the island hosts too many or too few natural predators. When a species colonizes a new area, the authors state that the species will either shift, expand or contract its realized niche. Chapter 6 is a theoretical exploration of dispersal models. The authors consider how insular stepping stones affect the dispersion of species, particularly, the effects that size and isolation of stepping stones have on dispersion. Further consideration is given to how dispersal curves and average distance traveled by pioneers impacts this study. In Chapter 7, the authors state that there are generally 3 consecutive phases to the evolution of populations after colonization. Initially, there is a trend for colonizers to evolve from R strategists into K strategists. The founder effect may also influence colonizing populations during this first phase. The second phase is marked by long-term adaptations to the local environment. In this period, abilities for dispersal are commonly reduced, and colonizers will either differentiate or assimilate with competing species. In the third phase, the evolution of colonizing populations may result in speciation and/or adaptive radiation. Field testing. The insular biota equilibrium theory was experimentally tested by E.O. Wilson and his then-graduate student Daniel Simberloff in 6 small mangrove islands in the Florida Keys. The islands were fumigated to clear the arthropod populations. Immigration of species onto the island was then observed in a first and second-year census. Wilson and Simberloff confirmed that there was an inverse relationship between the number of species on an island and the distance to the source region as predicted in the theory of island biogeography. 5. Impact. Applications. MacArthur and Wilson's theory of island biogeography has been widely applied outside of island ecosystems. For microbiota, the theory has been applied to the distribution of ectomycorrhizal fungi on trees, 6. The distribution of bacteria in water-filled tree holes, 7. And the distribution of fungi among shrubs. 8. While for flora and fauna, the theory's predictions have been realized with the species richness of plants on mountains, 9. And with the species richness of aquatic snails in bodies of water. 10. Novel applications looked at plants as islands for insect species, 11. And the dependence of the species richness of mites on the areas of the host ranges of rodent species. 12. MacArthur and Wilson's work has been used as a basis in other ecological theories, notably the unified neutral theory of biodiversity, 13. And has been foundational for the fields of landscape ecology, invasion biology, and conservation biology. 3. 1. Criticism. Several studies have disputed the underlying assumptions in MacArthur and Wilson's theory of island biogeography, specifically, the interchangeability of species in islands, the independence between immigration and extinction, and the insignificance of non-equilibrial processes. 14. The island biogeography theory can also be applied to habitat fragmentation. 15. However, limitations and nuances like edge effects, matrix effects, and community-level changes inhibit this theory from being universally applied to all systems. 15. In the 2001 preface, Wilson stated that, the flaws of the book lie in its oversimplification and incompleteness. 1. Legacy. In 2007, a symposium was held at Harvard University honoring the 40th anniversary of the theory of island biogeography. 3. Following this conference, a collection of papers was published in the book The Theory of Island Biogeography Revisited. 3. The SLOS debate is based on the author's suggestion that a single large reserve was preferable to several small reserves. Insular biogeography. Insular biogeography, 1, or island biogeography is a field within biogeography that examines the factors that affect the species richness and diversification of isolated natural communities. The theory was originally developed to explain the pattern of the species-area relationship occurring in oceanic islands. Under either name it is now used in reference to any ecosystem, present or past, 2, that is isolated due to being surrounded by unlike ecosystems, and has been extended to mountain peaks, seamounts, oases, fragmented forests, and even natural habitats isolated by human land development. The field was started in the 1960s by the ecologists Robert H. MacArthur and E.O. Wilson, 3, who coined the term island biogeography in their inaugural contribution to Princeton's monograph and population biology series, which attempted to predict the number of species that would exist on a newly created island. Definitions. For biogeographical purposes, an insular environment or island is any area of habitat suitable for a specific ecosystem, surrounded by an expanse of unsuitable habitat. While this may be a traditional island, a mass of land surrounded by water, the term may also be applied to many non-traditional islands, such as the peaks of mountains, 1, isolated springs or lakes, 4, and non-contiguous woodlands. 2. The concept is often applied to natural habitats surrounded by human-altered landscapes, such as expanses of grassland surrounded by highways or housing tracts, 5, and national parks. 6. Additionally, what is an insular for one organism may not be so for others. Some organisms located on mountaintops may also be found in the valleys, while others may be restricted to the peaks. 7. Theory. The theory of insular biogeography proposes that the number of species found in an undisturbed insular environment, island, is determined by immigration and extinction. And further, that the isolated populations may follow different evolutionary routes, as shown by Darwin's observation of finches in the Galapagos Islands. Immigration and emigration are affected by the distance of an island from a source of colonists' distance effect. Usually this source is the mainland, but it can also be other islands. Islands that are more isolated are less likely to receive immigrants than islands that are less isolated. This diagram shows the influence that the size of an island and its distance from the mainland has on the amount of immigration and species richness of the island. It can be seen that a larger island close to the mainland has the most species richness and a smaller one far from the mainland has the least. The rate of extinction once a species manages to colonize an island is affected by island size. This is the species area curve or effect. Larger islands contain larger habitat areas and opportunities for more different varieties of habitat. Larger habitat size reduces the probability of extinction due to chance events. Habitat heterogeneity increases the number of species that will be successful after immigration. Over time, the countervailing forces of extinction and immigration result in an equilibrium level of species richness. This diagram shows the effect of an island's distance from the mainland on the amount of species richness. The sizes of the two islands are approximately the same. Island 1 receives more random dispersion of organisms, while island number 2, since it is farther away, receives less random dispersion of organisms. This diagram shows the effect of an island's size on the amount of species richness. The diagram shows two islands equidistant from the mainland. Island 1 receives less random dispersion of organisms, while island 2 receives more of the arrows and therefore more random dispersion of organisms. Modifications. In addition to having an effect on immigration rates, isolation can also affect extinction rates. Populations on islands that are less isolated are less likely to go extinct because individuals from the source population and other islands can immigrate and rescue the population from extinction. This is known as the rescue effect. In addition to having an effect on extinction, island size can also affect immigration rates. Species may actively target larger islands for their greater number of resources and available niches, or larger islands may accumulate more species by chance just because they are larger. This is the target effect. Influencing factors. Total number of reptilian and amphibian species on 7 small and large islands in the West Indies. Degree of isolation, distance to nearest neighbor, and mainland. Length of isolation, time. Size of island, larger area usually facilitates greater diversity, the habitat suitability which includes climate, tropical versus Arctic, humid versus arid, variability, etc. Initial plant and animal composition if previously attached to a larger landmass, e.g. marsupials, primates. The current species composition. Location relative to ocean currents influences nutrient, fish, bird, and seed flow patterns. Location relative to dust flow influences nutrients. 8. Serendipity, the impacts of chance arrivals. Human activity. Species area relationships. Species area relationships show the relationship between a given area and the species richness within that area. This concept comes from the theory of island biogeography, and is well illustrated on islands because they are relatively isolated. 9. Thus, the immigrating species and the species going extinct from an island are more limited and therefore easier to keep track of. It is expected that as the area and species richness relationship are directly proportional to one another. For example, as the area of a series of islands increase, there is a direct relationship to the increasing species richness of primary producers. It is important to consider that island species area relationships will behave somewhat differently than mainland species area relationships, however the connections between the two can still prove to be useful. The species area relationship equation is 10. An example of what a species area relationship may look like when graphed. In this equation, represents the measure of diversity of a species, for example, the number of species, and is a constant representing the y-intercept. Represents the area of the island or space that is being examined and represents the slope of the area curve. 11. This function can also be expressed as a logarithmic function. 10. This expression of the function allows for the function to be drawn as a linear function. However, the core meaning of the function is the same. The area of the island dictates the species area relationship. Historical record. The theory can be studied through the fossils, which provide a record of life on Earth. 300 million years ago, Europe and North America lay on the equator and were covered by steamy tropical rainforests. Climate change devastated these tropical rainforests during the Carboniferous period, and as the climate grew drier, rainforests fragmented. Shrunken islands of forest were uninhabitable for amphibians but were well suited to reptiles, which became more diverse and even varied their diet in the rapidly changing environment. This Carboniferous rainforest collapse event triggered an evolutionary burst among reptiles. 2. Research experiments. The theory of island biogeography was experimentally tested by E.O. Wilson and his student Daniel Simberloff in the mangrove islands in the Florida Keys. 12. Species richness on several small mangroves islands were surveyed. The islands were fumigated with methyl bromide to clear their arthropod communities. Following fumigation, the immigration of species onto the islands was monitored. Within a year the islands had been recolonized to pre-fumigation levels. However, Simberloff and Wilson contended this final species richness was oscillating in quasi-equilibrium. Islands closer to the mainland recovered faster as predicted by the theory of island biogeography. The effect of island size was not tested, since all islands were of approximately equal size. Research conducted at the Rainforest Research Station on Barrow, Colorado Island has yielded a large number of publications concerning the ecological changes following the formation of islands, such as local extinction of large predators and the subsequent changes in prey populations. 13. Applications to island-like systems, ILS. The theory of island biogeography was originally used to study oceanic islands, but those concepts can be extrapolated to other areas of study. Island species dynamics give information about how species move and interact within island-like systems, ILS. Rather than an actual island, ILS are primarily defined by their isolation within an ecosystem. In the case of an island, the area referred to as the matrix is usually the body of water surrounding it. The mainland is often the nearest non-island piece of land. Similarly, in an ILS the mainland is the source of immigrating species, however the matrix is far more varied. By imagining how different types of isolated ecosystems, for example a pond that is surrounded by land, are similar to an island ecosystems, it can be understood how theories and phenomena that are true of island ecosystems can be applied to ILS. 14. However, the overall immigration and extinction patterns that are outlined in the theory of island biogeography as they play out on islands, also play out between ecosystems on the mainland. 15. The concepts of area of an island and the level of isolation from a mainland as presented in the theory of island biogeography apply to ILS. The main difference is in the dynamics of area and isolation. For example, an ILS may have a changing area because of seasons, which may impact its degree of isolation. Resource availability plays an important role in the conditions that an island is under. This is another factor that changes in ILS in comparison to real islands, since generally there is a greater resource availability in some ILS than true islands. 14. Species area relationships, as described above, can be applied to island-like systems, ILS, as well. It is typically observed that as the area of an ecosystem increases, the species richness is directly proportional. One major difference is that values are generally lower for ILSs than true islands. Furthermore, values also vary between true islands and ILS, and within types of ILS. 14. Applications in conservation biology. Within a few years of the publishing of the theory, its potential application to the field of conservation biology had been realized and was being vigorously debated in ecological circles. 16. The idea that reserves and national parks formed islands inside human-altered landscapes, habitat fragmentation, and that these reserves could lose species as they relaxed towards equilibrium, that is, they would lose species as they achieve their new equilibrium number, known as ecosystem decay, caused a great deal of concern. This is particularly true when conserving larger species which tend to have larger ranges. A study by William Newmark, published in the journal Nature and reported in the New York Times, showed a strong correlation between the size of a protected U.S. national park and the number of species of mammals. This led to the debate known as single large or several small sloths, described by writer David Quammen in The Song of the Dodo as ecology's own genteel version of trench warfare. 17. In the years after the publication of Wilson and Simberloff's papers, ecologists had found more examples of the species-area relationship, and conservation planning was taking the view that the one large reserve could hold more species than several smaller reserves, and that larger reserves should be the norm in reserve design. This view was in particular championed by Jared Diamond. This led to concern by other ecologists, including Dan Simberloff, who considered this to be an unproven oversimplification that would damage conservation efforts. Habitat diversity was as or more important than size in determining the number of species protected. Island biogeography theory also led to the development of wildlife corridors as a conservation tool to increase connectivity between habitat islands. Wildlife corridors can increase the movement of species between parks and reserves and therefore increase the number of species that can be supported, but they can also allow for the spread of disease and pathogens between populations, complicating the simple prescription of connectivity being good for biodiversity. In species diversity, island biogeography most describes allopatric speciation. Allopatric speciation is where new gene pools arise out of natural selection in isolated gene pools. Island biogeography is also useful in considering sympatric speciation, the idea of different species arising from one ancestral species in the same area. Interbreeding between the two differently adapted species would prevent speciation, but in some species, sympatric speciation appears to have occurred. Insular biogeography. Insular biogeography, or island biogeography, is a field within biogeography that examines the factors that affect the species richness and diversification of isolated natural communities. The theory was originally developed to explain the pattern of the species-area relationship occurring in oceanic islands. Under either name, it is now used in reference to any ecosystem, present or past too, that is isolated due to being surrounded by unlike ecosystems, and has been extended to mountain peaks, seamounts, oases, fragmented forests, and even natural habitats isolated by human land development. The field was started in the 1960s by the ecologists Robert H. MacArthur and E.O. Wilson, who coined the term island biogeography in their inaugural contribution to Princeton's monograph and population biology series, which attempted to predict the number of species that would exist on a newly created island. Definitions. For biogeographical purposes, an insular environment or island is any area of habitat suitable for a specific ecosystem, surrounded by an expanse of unsuitable habitat. While this may be a traditional island, a mass of land surrounded by water, the term may also be applied to many non-traditional islands, such as the peaks of mountains, 1. Isolated springs or lakes, 4. And non-contiguous woodlands. 2. The concept is often applied to natural habitats surrounded by human-altered landscapes, such as expanses of grassland surrounded by highways or housing tracts, 5. And national parks. 6. Additionally, what is an insular for one organism may not be so for others. Some organisms located on mountaintops may also be found in the valleys, while others may be restricted to the peaks. 7. Theory. The theory of insular biogeography proposes that the number of species found in an undisturbed insular environment, island, is determined by immigration and extinction. And further, that the isolated populations may follow different evolutionary routes, as shown by Darwin's observation of finches in the Galapagos Islands. Immigration and emigration are affected by the distance of an island from a source of colonists' distance effect. Usually this source is the mainland, but it can also be other islands. Islands that are more isolated are less likely to receive immigrants than islands that are less isolated. This diagram this and further, that the isolated populations may follow different evolutionary routes, as shown by Darwin's observation of finches in the Galapagos Islands. Immigration and emigration are affected by the distance of an island from a source of colonists' distance effect. Usually this source is the mainland, but it can also be other islands. Islands that are more isolated are